Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome back, everyone, to another exciting week at the Genetics Podcast. I'm very excited to be here today with Dr. Xin Jin, who's a pioneering neuroscientist and associate professor at the Scripps Research Institute in beautiful, sunny San Diego or close by. So Xin develops and applies techniques that help us to connect the dots, in my view, between what's happening on a cellular level and what's happening on an organismal and disease level. One of the very powerful ones that's come out relatively recently is in vivo Perturb-seq that we're going to talk about today. And Xin's work is really opening up powerful ways to connect what's happening from a genetic risk perspective on a cellular level all the way through to common diseases like autism and other neurogenetic diseases. But I think this method can be applied across the whole spectrum of biology, right? So welcome and thanks for joining us, and look forward to exploring this today.
Thanks, Patrick. It's nice to be here. I look forward to our conversation.
I would love to start with this method that I spoke about in vivo Perturb-seq. Could you walk everyone who's not familiar with it already with how it works and what kind of avenues it opens up from a scientific perspective?
Yeah, definitely. You put it really beautifully, right? There are tremendous amount of knowledge in the field of genetics genomics that we have, collectively as a field in the last, 2 or 3 decades, thanks to these increasingly powerful and inexpensive genomic tools. Now we know many devastating disease and disorders are actually has a components of genetic molecular mechanism, starting from neurodevelopmental disorder all the way to devastating adult onset disorders, and that impact collectively 1 in 3 adults in America, that severe level of these mental illnesses. However, just because we know what kind of genes are implicated or make us vulnerable to a disease, it doesn't really mean that we understand or know how to treat them. In fact, the majority of these disorders has no effective treatment at all. And now the question is, we understand the gene, and many of them are well studied in many different animal models or cellular models. However, disease is this emergent properties we like to think. It's not one gene or one molecule that goes awry. It is a collection of many different, sometimes combination of these mutations or variants we like to call. And therefore, to be able to appreciate them in a collective organismal level or at least cellular level, and try to start asking questions such as, how much heterogeneity are there when we're talking about something as complex as Alzheimer or autism spectrum disorder? Is it one kind, or is it 20 genes telling you 20 different disorders? And if it's the latter, we need to actually know sooner than later, because there's no unlikely to have one drug, one treatment for the huge spectrum of disorder. And I think this is really where how we came up with this idea of using these systematic genetic approaches to look broadly across a spectrum of disorder, a spectrum of long risk genes, perturb them one at a time, but also in parallel, so that we are not taking decades to solve all of the genes, and also appreciate them that every single gene and genetic variants might impact different cell type differently. By combining these 2 technology, mainly is CRISPR as well as single cell genomics, we hope to be able to take a long list of risk genes. We no longer have to choose, but look at them, and there are lots of function action across a broad spectrum of cell types or even organ type down the road. That can help us to realize, these 5 out of these 200 genes seems to be always acting the same cell type. And their loss of function leads to the cell type goes awry in the same way. And maybe we can cluster them into a subtype, put them in a bucket, and we can study them more in depth. And now we know there's a better, let's say, biomarkers or phenotypic readout we can use as a handle to understand their mechanism or the reversal possibility using other type of rescue means. And I think that will be really the hope that we start with the almost forward genetic way to look for mechanisms and looking for convergence, and then figure out ways to understand the mechanism underlying these complex disorders, and then in hope to be able to reverse some of them.
Yeah, it's amazing, and we're going to talk a little bit about autism genetics as a model for this because you do a ton of work there. I just want to recommend to everyone episode 188 of the podcast pretty recently. We had Dan Geschwind on and talked about autism as it's obviously a spectrum. We all know that, but genetically speaking, we have rare and ultra rare neurodevelopmental disorders where it shows up, and we also have obviously more common phenotypic trait in the population. Maybe we could zoom into that as an example because there are literally thousands of genes that show up from polygenic risk scores where they're very subtle effects through to monogenic disease genes. And maybe you can talk a little bit about how you go from that starting point to better understanding. And there is, by the way, a beautiful video that you and your team have put together that we'll make sure we include in the show notes that people can watch that gives a visual overview of this as well that I think is really helpful. So we'll make sure that gets included.
Yeah, definitely. I'm not a geneticist, but I work very closely with many fantastic geneticists, Dan himself included. And I think it's really amazing, right, to see in the past, I want to say literally maybe a little bit over 10 years, we have taken maybe one of the most complex, I would say, disorder that is not monogenic, has many different genetic contributions, but cracking this puzzle. So as you said, there are many different kind of genetic architecture, the sort of so-called polygenic, meaning that you have a lot of variants, and each of them probably contribute very little, but in combination, they probably shifted the vulnerability or resilience population. But each of them will be very difficult to study because they often are, small effect size or weaker penetrance. And on the other spectrum, you have this really interesting, we call de novo variants, and then the geneticists primarily looking for sort of within the trio of family, instead of taking the GWAS approach, which is autism and control, and looking for the difference. That is what the GWAS typically look for. In the de novo variants, they typically take a family of the trios, and of course, many, many of them, and looking for between the affected child versus the unaffected sibling or the family members, what are the de novo variants that's not present in the healthy siblings, but present in the affected child? And in this case, the chances that you'll find these really, really rare variants is approaching zero. And therefore, if you found one, it's very likely that this is actually the causal variant. And there are a lot more sophisticated sort of analysis that's going on top of it. As you can see, this needs a lot of power. So I think when the first batch of paper came out, people started hypothesizing maybe de novo variants a way to think about this disorder. And in fact, like I think it was the 2012, 2013, a few back-to-back papers show that these variants truly can be found. And they found a few, I think, at a level of sort of like single digit, about 8 to 10. And that time you have so little data, data quality wasn't so great. But the amazing thing is, in those papers, that the 10 variants they found was exactly the top 10 variants we have today. If you rank all of them sort of based on effect size or the likelihood, explain population of autism patients. So it's really telling us that, of course, with more and more data, the tail will keep growing, right? You will see a few more genes with a borderline statistic now jumping into significance as the sequencing getting better. But I would say the other way to think about it is always the distribution. The top, I want to say 50 to 100 genes, that's loss of function, causally, statistically saying causally linked with autism and developmental delay, have been really remarkably reproducible across cohort of patient, across different type of statistical analysis, and that is probably one of the most amazing puzzle in neuroscience as well as in population genetics, that we have now this list of genes and genetic knowledge we didn't have before.
And I learned from the video that I encourage everybody to watch that the way you're doing this is you're making edits effectively in mouse in mice. These are being distributed across cell types, across the brain, for example. And then you can map those networks. You do single cell sequencing, and you can, I suppose, also do RNA and other kinds of any single cell technique. In theory, you could profile those, and you get a massive readout that's effectively probably a matrix of the genetic change that has been made, the cell type, and then all the different outputs. And so I'm curious to get just a sense of the scale and scope of the data. Like when in a particular experiment, how long does it take to set one of these up, and what comes out the other side, just so people can wrap their heads around the kind of data that could be generated through a method like this?
Yeah, no, that's a great question. So in terms of the scale, I want to argue that now doing things in vivo, obviously you are signing up for many different cell types, right? Like compared
Yeah.
to what you could grow on the petri dish in vitro. Like typically in vitro, there are very complex cell types such as immune cells, right? But it will be at a scale of sort of like no sort of a continuum of less inflamed and more inflamed. And this is something you can push in vitro, adding this kind of stimulant. But in vivo, even in the mouse brain that we're talking about, according to just the paper that Alan Van Atlas has mapped last year, arguably there's about over 200 superclass and over 5,000 different cell types, right? Like
Wow.
depending on your merger or splitter. So now you have a village of so many different cell types, and I guess brain is one of sort of the most amazing organ for that reason, but the question really is when something as complex as autism comes in, and let's say 100 different mutation that leads to this, little black box of the brain, and then output is some sort of disorder states, right? The question is, are any of these 50 mutation doing the same thing or different thing? And also in the middle black box full of 5,000 different cell type, what cell types are more vulnerable than others? It's possible that it's these like one or two cell type out of the 5,000 that actually explain majority of the mutation, or is it very distributed, sort of very sparse way of thinking about the disorder? That's sort of what we're really hoping to gather. And exactly like you said, what we are doing is to be able to introduce these perturbations across the whole brain, although there are certain sort of brain regions are more implicated in autism than others. But what we can do is to make sure that majority of the cells, that one cell received one mutation or one perturbation. And we do this purely in vivo. So the animals growing, the cells are still maintaining their,
Yeah.
psychoarchitecture, talking to the rest of the brain, except they have that gene mutated. And days or weeks or even months after, it doesn't really matter because it's in vivo, right? You don't have to even change the media like what you would have to do in
Right.
vitro. Very happy. And we can extract these perturbed cell because they were labeled by the blue fluorophore. And then you can use single cell multiomics or genomic methods to start assaying, measuring as many modality from the single cell as possible to be able to ask when this mutation happened. And I know this is a cell type of astrocyte. What happened? Did they go away? Did they change? Did they not care when the mutation happened? And you can go in so forth and build in this really big matrix of all the perturbation across all the cell types. And I would say traditionally, if you're interested in sort of talking about scale, right? Like if you're interested in 100 mutation, you have no choice but to make 100 knockout cell line or knockout mouse lines and then analyze them in triplicates. And, that's very expensive and time consuming, whereas in vivo Perturb-seq can bring all of them into relatively few number of animals by using single cell as a measurement unit that not only allow you to do sort of more perturbation sooner and quicker and less batch effect, but I want to argue that they also give you the resolution that you wouldn't easily have because now we know, again, the 5,000 cell types in the brain, some are more important than others. What are the cell type that's more vulnerable than others? This is what we're really hoping to find out.
Have you or others applied this to other neurogenetic disorders? I'm thinking dementia, frontotemporal dementia, Parkinson's, ALS. There's probably huge just in the brain alone, a huge amount you can learn here. And yeah, I'm curious to learn about that.
Yeah, we started with neurodevelopmental disorder in part just because working with fantastic geneticists, right? Like we started a few years ago. So we're not talking about the genetics we know today, but rather almost probably 6 or 7 years ago. And that's how we choose sort of, genetic problem autism is fantastic. But you're right that there are increasing amount of knowledge of genetic sort of vulnerabilities in other neural disorder as well. So we're working towards a few others, especially in the neurodegenerative space, just because I think it's very hard to convince a neuron sitting on the petri dish to age.
Right.
Whereas
Yeah.
in the age mouse brain, you can say, hey, mouse brain is 2 years old, human different. But at least you will have this intact physiology, the neural inflammation, the really
Yeah.
complex sort of interoception cues that an aged animal is an aged animal. It's not just because the brain is sitting in the dish for 2 years, but rather because the entire organ and the metabolism changes, right? Which is what in vivo and these kind of intact physiology strength really is. So we're excited to find out, but obviously working with aged animals are more challenging. So stay tuned. Hopefully I'll be able
Yeah.
to talk about it soon.
Yeah, I get it has me thinking that I guess earlier onset diseases are a little bit simpler just due to to the timescales we're talking about. These are mice, not petri dishes or flies or something else. But are there other moving outside of the brain? Are there other organ systems that make natural sense for this to you, where you could see it helping pretty quickly?
Yeah, I think there are many, organs that are fascinating. I'm a neuroscientist, of course. I love the brain, but I think the concept of you being able to do in vivo screening at large scale is really what I hope that I can convince more people in the field to start adopting and use, right? Many biologists in general, we're very used to thinking about cellular system as a model. I think there are at least two reasons. One is that you can grow them on petri dish at scale very easily, right? Like a dish with 10 million cells, you can do 10 million single experiment and things like that. But I want to argue that in vivo, we have just as many cells. And the entire mouse brain is about 100 million cells. We're not in shortage of cells. But what's
Mm,
really different is that in vitro, you can do transfection. You can introduce whatever transgene you want, like the amazing molecular biology, right? You can edit genome, you can put in GChem there. You can do this in 2 days, right? Like
yeah.
mix it with lipophilic. Whereas in vivo delivery has been much harder. So just because you have the entire 100 million cells in the mouse brain doesn't mean you can perturb them and label them easily.
Right.
So that's one of the reason that recently we did AAV engineering and really help accelerate the labeling and perturbation efficiency. Because if your labeling efficiency, let's say, increase by 20x increase of the scale, it means that you can do 20x less experiment, or you can do the same amount of experiment and get 20x more data. And the latter of which is probably what we're going for. And I think the similar, effort has been really happening in parallel in other organs as well, for liver, for heart. These kind of labeling technology using various different viral vectors are getting really mature. So I think these organs are fantastic starting point to start asking ourselves, well, is in vivo screening actually a sort of a more suitable way and likely to replace some of the in vitro systems. Another thing is cell type diversity, which is, a
Yeah.
blessing and a curse at the same time. In vitro cell culture, typically, even the 3D cultures are getting sophisticated over time, right? But still, we're talking about probably less than maybe a dozen cell type diversity. And for some organ, that might be just enough. It's already really great to mimic the pathology. But I think in vivo, the cell type diversity is shocking, right? With all the immune cell infiltrating and in and out. So I think the question is, what is the right model for what kind of question that people are interested in? And if we can start doing more and more in vivo screening, starting as a parallel screen with in vitro, how much of the insight we learn from in vivo and in vitro are overlapping? How much of them are, shared across models?
Yeah.
None of the models, right? Like some are more useful. So I think that's probably an interesting time that we can start asking ourselves what type of in vivo joint modeling is helpful, because ultimately, we're not in love with mice, we're not in love with petri dish, right? Like what we really care about is this disease or disorder we're trying to treat, or whatever model that can help us to get through that. It will be helpful for the field to know.
Yeah, it seems like the in vivo context and the variety of cell types feels like you have a on the one end of the spectrum, you, as you spoke about before, you can do a knockout mouse and you have the whole organism, and you can do cellular models, but this is something in between where you get the scale and variety, but you also get the in vivo context, and it feels like there's got to be useful information there that we're missing on either of those extremes.
Right. The question is, what is really the biomarker, or what is really the feature that is disease driving, let's say, or disease modifying, that we are trying to model. And when we know that, right, and I think the goal of Perturb-seq and the goal of all the modeling is to capture that. Once we have that as a reliable marker, and then we can start thinking about ways to modifying it.
Yeah.
But starting from genetics, I think the most important is to get to the mechanism, and we need to use all the models out there that can be helpful.
How many gene targets or variant targets, I guess, are you typically putting in an experiment? Is it hundreds or thousands or millions? Like what's the right level for that?
Right. I mean, if we're just talking about loss of function, sort of knocking out a gene, right? Like in total, there's like about 20,000 genes you can knock out. Like that's
Yeah.
kind of I consider as a lot upper limit. And then some of them, if they are not expressing, it's not worth knocking out, right? Like, so I think the space at the subgenome scale, like a couple thousand, is what that's very doable. Really depends on the context. If all you care about is a very small subpopulation that matters, of course, it's much harder. But if you just want to like collect enough information, and I think doing this at the subgenome scale is doable nowadays.
Yeah, like the thing I'm thinking about is taking, and this is maybe extending into the neurodegenerative case, but you have thousands of variants or approaching millions in some of the polygenic risk scores, and wanting to unpick like I think there's
Right.
emerging evidence in a lot of fields that the polygenic risk scores tend to collapse on particular pathways, right? Like you don't actually have 1,000 different pathways. You've got five or six or seven that matter, and different variants collapse on different ones. And this strikes me as the kind of thing where you could introduce all of those and then look at what's going on in each individual cell type and try to figure out like what is the you I love the way you described it as an emergent property, but what is the
Right.
the higher level pathway signal that we're getting from this variant, and how is it propagating through the system versus it's like, it is just this variant on an island by itself.
Yeah, and we learned that from sort of our non-neuroscience community as well, right? Sometimes the primary mutation or the gene that drive the disease may not be the drug target. Like they might be
Yeah.
too hard to drug or just not druggable for all the reasons, right? But like the propagating network, as you said, is usually the place that the therapeutics happen. And I think we're probably falling through the same camp, hopefully at the end of this tunnel when we're figuring out how this diverse autism give rise to this complex disorder. But I would also argue that, like it's all about gene ranking. Like inherently, some genes are more important than other. You can use the
Yeah.
genetics from human data to rank them. You can use the pathways to rank them. And there are a lot of very exciting causal inference that people are also doing just because you observe, in the differential gene expression way, these genes go up, these genes go down, but they don't go up and down for the same reason. Some are drivers, some are followers, right? Like how do we figure out who's the driver? Then maybe driver is more worth perturbing and understanding than the follower because they're downstream, right? So I think really married to these in parallel growing efforts in bioinformatics and machine learning, and these large models. And I think it would be really cool for us to use them to leverage these sophisticated tools that traditional biostatistics are harder to give this kind of level of insights because the amount of data we have is surpassing the sort of the infrastructure, and then use that to really cleverly nominate, even if we're gonna do sort of thousands, but what are the top 100 we should do first, right? And using that to lead to insights, let's say lead to even predictions. And we can do this lab in the loop very quickly because the prediction can be immediately tested through another
Right.
round of experiment. And at some point, we may be able to have a really good classifier or predictor or, whisper to the cell, whisper to the perturbation, whatever you name it, and with a reasonable sort of prediction score. And I think this is obviously a very daunting task just because there's so many genes and so many cell type. And this is maybe a higher bar of these efforts nowadays trying to predict perturbation and in unseen cell type or unseen mutations. But I think we're going to get there especially with high quality data and this kind of infrastructure for us to understand what are the most important genetics, to understand what are the most, urgent prediction that will be rapidly tested in the loop fashion.
How hard is it to make the leap from mouse to human in these cases? Is that a big bottleneck, or how much are these pathways preserved enough that that gene ranking is preserved or the signal is preserved in some way?
That's right. That's a great question. So for autism spectrum disorder, neurodevelopmental delay, a lot of these genes encode transcription factor and synaptic molecule. At a gene level, they code totally conserve. Like very
Yeah.
rarely, I'll find even a gene symbol that is different between the two different kind of species. But you are right that is it possible that there are subtle differences for, let's say, a given transcription factor and their cell type implication in mouse versus human. So I think we have ongoing project with collaboration with many experts in the field. And the idea is to be able to just test them all and find out, right? And I think for transcription factor, you can imagine that evolution bringing different cell type or different cell type regulation just because human brain is very different from mouse brain. But that doesn't mean that the way that a loss of function of a transcription factor shouldn't be rescued the same way, right? Like inherently, there are molecular mechanisms that can be conserved. We are testing these same list of genes across a variety of different model and just trying to sort of, for the first time, have a fair comparison and then see,
Yeah.
hey, across all the models, what are the things that we see that are similar? What are the things that's different? For the things that's similar or different, which are the one that matters more, right? Like if not because they are differential gene expression, meaning there's a function, it's literally a calling card for us to dive in deeper into the mechanism. But I think considering the model differences, considering the genetics, the field will help us to, collectively move forward thinking about deeply of what these disorders really are and what model
Yeah.
is most reliable or feasible model.
Yeah, that following on from that, how do you see this? How do you see this having the biggest impact on therapy development? I'm curious to see what you think is going to be the big impact there.
Right. And I think, there are a lot of very exciting progress in the field of antisense oligo gene therapy, especially for these rare, devastating diseases and disorders. So if you're thinking about 100 different mutations and designing 100 different sort of sets of antisense oligo or CRISPR therapies or different ways to modulate gene expression is obviously possible, but they're very slow and painful and challenging just because you expose the risk to 100 different potential off targets and things like that.
Yeah.
Right? But if, let's say, we will be able to figure out a point of convergence, meaning that I'm not saying singularity. I'm saying potentially these 100 different genes that give rise to sort of loss of function, leads to vulnerability in developmental disorder. But maybe 5 or 6 of them are occurring in the same cell type that need to be rescued around the same time in the same transcription factor complex. And maybe instead of developing 5 independent antisense oligo, CRISPR therapy, you name it, we may be able to lump sum them together and develop one potentially downstream network sort of therapeutic target, like what you mentioned. Or maybe it's possible that the target is one of these 5 genes, but
Yeah.
that's likely the driver. And I think that's a very practical and sort of very impactful way that I can start thinking about that can greatly accelerate the therapeutic development and piggybacking on all the efforts that the field's already building towards.
Yeah, and is there a potential to use it almost as a synthetic rescue where you have the knock-in mouse with the disease, and then you basically do the reverse, right? Where you make a bunch of edits that you hypothesize could rescue it, and then you look at the cell types where the network is rescued in some way.
Yeah. No, that's a really, really great question. We think about this all the time, right? Like rescue experiments, the sufficiency experiments always
Yeah.
much harder. And it's also tricky. This is delving into sort of the differences of the genetics, right? Like we already know from great work from geneticists in the field, Huda, Zombie, and many others identify MECP2. And it's one of the genes that it just has to be just right. If it's overexpressed,
Hmm.
it's intellectual disability. If it's, like haploinsufficient, even losing one copy, that is autism or
Yeah.
Rett syndrome and so on, right? So I think rescuing and titrating to the right level matter, and this is the observation from molecular genetics, but I think it just says that different genes and their sensitivity or haploinsufficiency is very different. Being able to rescue them to the right way, but both from experimental level, like what you mentioned, sort of doing a secondary screen, or this sort of therapeutic means of bring them back into right balance is challenging, and it's obviously depending on the gene. So if we're gonna do that experiment, we need to, figure out that founder knocking gene very carefully. But that's definitely something we're also thinking.
Amazing. I want to transition a little bit to how you got to where you are today. I think you grew up in China. Your grandfather worked as a plant taxonomist, and this had a significant influence on you. I'd love to hear a little bit more about that. It's interesting how our childhood experiences can reverberate through the rest of our life in terms of what we choose to do.
Yeah, I grew up in the, in Research Institute, which is like a botanic garden,
Oh,
that
cool.
my grandfather works in. And then for me, it's probably the earliest, this as early as like, you can get, I guess, inspiration of being a scientist and what's it's like and how fun it is. And for me as a child, just wandering around the botanic garden and making friends with all the trees and different plants. And this is sort of in the southeast part of China, climate-wise, very similar to San Diego. This feels like
Yeah.
home. With just like marvelous amounts of plants. And then as a botanist, I can imagine like my grandfather's job back then from a child's eye is very romantic because what you do is you're going around and you take this notebook and there's no iPad then, right? Like you
Yeah.
make a beautiful drawing of different plants and you take notes about what they are. And I think that has a little bit of a genetics influence. So I learn a bit about, oh, there are different species and genre, things like that. But most importantly, what fascinates me is that, my grandfather used to show me sort of this is the poisonous plants, don't touch it. And
Right.
this one is a beneficial herb, whatever it is. And they look very similar. And when I was very young, I'm always just confused about sort of what is the form and what is the function. They look very similar, but at what
Mm.
level they're different. Okay, molecular level. Then what is molecule? What is chemistry? So that's a little bit sort of a top-down way to think about biology, that things that seemingly extremely similar from one level at a morphological level can be so different when you're accidentally chewing them and getting them into
Yeah.
your body, onto your skin. So that was a very sort of stark contrast, almost like instinctual learning, I guess, when you're a
Mm.
child. And that got me very excited about biology in general, but more sort of the molecular and chemical basis of life.
Yeah, that's fascinating. And it is I mean, do you have any of his old notebooks or anything like that? I imagine those must be incredibly nostalgic if you do.
They are so pretty. I'm barely able to draw, but he's a marvelous bookkeeper, and there are really beautiful notebook that I still kept with me.
Yeah, that's wonderful. And when you were 18, you moved, I understand, from China to the US to go to MIT. What was that like? How did that shape you? Because I first of all, it's an incredibly hard place to get into as an 18-year-old. So you must have been doing some amazing work to stand out and go there. But also, you move halfway around the world, completely different environment, known for its intensity, I think, and what what a smart group of people it is. So I'd love to just hear what that was like and what that felt like to move there at age 18. And I think you've been in the US ever since, right?
That's right. Yeah. I mean, when you're young, you do certain things without knowing fully the consequence.
Yeah.
What I feel like right now, the way we describe it, I was like, yeah, totally right. Why would I do that?
Yeah.
But I was always young. I was rebellious. I just want to be far away from home. And that's part of sort of this,
Oh, interesting.
let's get out and go somewhere else and speak a different language. But yeah, I went to MIT for my undergrad, and that was in part because I was doing chemistry Olympiads, and I think that the Olympiad circle, there's always sort of people thinking about studying abroad, and
Yes.
that's how I first heard about it because my parents are direct not scientists, and then we don't really know anyone who study abroad, but that was a community, and that's also sort of my first social community. Coming to MIT, there are a lot of Olympiads there, and it's a really wonderful. I think about it very fondly, and some of my very early, I had my first manuscript when I was working in the lab, in the chemistry
Yeah.
lab at MIT in Alice Ting's lab. I was fortunate to work with great graduate students over there, and they helped me tremendously, not just scientific, but also just like how to be professional at a workplace. But that was my first real job, right?
Yeah.
How to communicate your science, how to ask for help, how to really work like a scientist. I think that's really a stark difference between someone who is naive and read about science and then I competed for a chemistry Olympiad, so I know what chemistry is versus doing research as a full-time job, and I loved it. So that was really interesting and important formative period, and I think still think about very fondly. I'm super grateful for all the opportunity I had there.
You mentioned the chemistry Olympiad, and so I can't resist this question. Just a couple of days ago, the top OpenAI model, I understand, won gold at the math Olympiad. I think only one human beat its score, is my understanding, and it had the same amount of time, had a lot more compute power, I suppose, than the human brain does. But I'd love to get your perspective on that, having done those kind of competitions. What do you think about the trajectory that these models are on, and how it changes or doesn't change the way that people at the top of the field work?
Yeah, no, I'm super excited to see where things unfold. But I would also say, and I keep telling, my friends in the ML world as well, that biology is different. I'm not trying to say like, oh, our
Yeah.
problem is more complex, but it is, right? Like, and you think about it, there are so much math and physics theorems and logics are written in natural languages, right? Like this is gravity and this is the
Mm.
formula. As long as you understand it, you can use it to solve problems and you can build on top of that because theorems are written. Unfortunately, or fortunately, as biologists, we don't have much theorems, right? Like we have the central dogma, which is really great,
Right.
but on top of that, there are a lot of modification already, right? There are RNA level microRNA regulation that wasn't quite there, and different cell type do it differently. So I think the challenge is just more daunting for biology.
Yeah.
But what we also do have is massive amount of data. Maybe a bit messy. Maybe everybody sequence things in different ways, and there are batch effects and things like that. But we do have massive amount of data, sequencing data from human population, genomic data from researchers like us, so on and so forth. So I think if we have such wonderful LLM taking the entire knowledge of the internet and then make sense out of math and physics and beat an IMO medalist,
Yeah.
how long would it take, or would it be possible at all to do the parallel in the biology field, take massive amount of data, but then you have to curate them and think about them in very intelligent way because I don't know if the way we think about language, a word before, a word after together has a meaning, but for two genes that's next to each other in the genome, they may or may not have, this is not usually how it works. So I feel
Yeah.
like there are probably new infrastructure, new way of thinking about it can happen, but I'm very optimistic and I'm very excited.
Yeah, and I think that methods like yours and what we spent a lot of today talking about, uh, figuring out how we close the loop quickly between experiment, prediction, and another experiment, there are there are hundreds and thousands of those probably that we need to go from fundamental biology understanding through to therapeutics and improving the healthcare system. And I think there's a lot of these small feedback loops that if we get better at closing them, then I can see, I don't think it's it's a foundation model that solves everything in biology overnight, but you could imagine it as a series of feedback loops that start to, if they all come together at the same time, can probably roll downhill pretty quickly.
I absolutely agree.
Well, thank you so much for taking the time today. Is there anything that people listening can help you with? Are you looking for collaborators? Are you looking for any if you're hiring PhDs, postdocs, anything like that? Then now is a great time to call it out.
Yeah, we're always actively hiring. So if any of the work that sounds interesting to you, please feel free to reach out to us.
Amazing. We'll put your lab website in the show notes. And thanks again for your time. And thanks everybody, as always, for listening. And we'll see you next time. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selected Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.